In this video, we're going to talk about a nucleic acid and a protein synthesis. So we're going to talk about what is nucleic acid is, what's the purpose of nucleic acid in our body, and how this nucleic acid helps in the protein synthesis in the body, in our body, or in any living organism. So let's start with the nucleic acid. What is nucleic acid? Nucleic acid are biomolecules that are normally involved in the transfer of genetic information from one cells to another cells. Or you can say nucleic acid are a biomolecule that normally store all the genetic information that is required by a living organism to sustain its life. They have all the information that is required by the living organism to do all its biological function. So there are two types of nucleic acid. One is called RNA, which is ribonucleic acid. Second is DNA, which is deoxyribonucleic acid. So RNA is normally found in the cytoplasm of the cell, and DNA is normally found in the nucleus of the cell or nuclei of the cell. So let me show you how the cells looks like. So this is cell. And within the cell you have a small area covered by a membrane. This is your cell membrane. This is nucleus. That's why we call as nuclear membrane. And then this is nucleus. And within nucleus you have DNA. And this area which is outside the nucleus, this entire area that is outside the nucleus, we call as cytoplasm. So RNA is present in cytoplasm. Right. So this is a basic structure of the cell that you need to know before you understand where is RNA is found in the cytoplasm and DNA is present in the nucleus. Right. So now let's talk about a special term called nucleotide. A nucleotide is a repeating structural unit or monomer of a polymeric nucleic acid. Right. So we're going to talk about this nucleotide in the next slide. Let's go to the next. So we'll talk about the composition of the nucleic acid, how nucleic acid is made up of. So we have a nucleic acid. Nucleic acid is made up of nucleotide. So nucleic acid is a polymer of polymers of nucleotide. When more than one or more than 10 nucleotides combine together, they form combine together in the form of polymer, which we normally call as polynucleotide. So polynucleotide is nucleic acid when many nucleotides join together. Right? And what is nucleotide? Nucleotide is made up of three parts. One is called base. Second one is called sugar. And the third one is called phosphate. So nucleotide is made up of three components: base, sugar, and phosphate. So base is made up of two parts. There are two types of base: pyrimidines and purines. And sugar can be ribose or 2-deoxyribose, and you have a phosphate. So this is the structure of nucleotide. So if you can see, you have a sugar, which is one of the component. Then you have a base, which is the first component. It can be purine and pyrimidines. Then you have the third component, phosphate. This is your phosphate. Right. So nucleotide is made up of all these three components together. Now let's talk about each of them separately. Let's talk about bases first. So bases are the nitrogen containing compound in nucleic acid that are two types, pyrimidines and purines. So if you can see, this is the structure of pyrimidines. This is a nitrogen containing compound. Then we have purines, again nitrogen containing compound. So what's the difference between pyrimidines and purines? Pyrimidines is a single cyclic compound, whereas purines is a fused ring compound. Here you have two rings fused together. Right. So this is how you can differentiate between pyrimidines and purines. Right. So in DNA, purine bases with double rings, means purines has two rings that I just told you about, right? Two fused rings. Where the pyrimidines has only one ring, nitrogen containing ring, right? So purine is of two types. You have two type of base of purine, which is A and G. A is your adenine, G is your guanine. Whereas pyrimidine bases with single rings are, there are two types, cytosine and thymine, right? And then you have one more type called uracil, U, right? So so you will find adenine, guanine, cytosine and thymine in DNA, right? And in RNA, second type of nucleic acid, this thymine is going to get replaced. This thymine is going to get replaced by uracil in RNA. So in RNA you don't have thymine, you have uracil instead. Every other thing is same, adenine, guanine, cytosine, but in RNA you don't have thymine, you have uracil. But in DNA you don't have uracil, you have thymine. So that's the difference between the bases in DNA and RNA. Right. So let's talk about the pyrimidines and purines one more time and see the structure. Like I already told you, pyrimidines has cytosine, uracil and thymine. Uracil is present in RNA only, wherein thymine is DNA only. So this is the main difference that I told you. Cytosine in both DNA as well as RNA. So these are the structure of the cytosine, uracil and thymine. And for purine you have adenine and guanine. These are present in both DNA and RNA, and this is the two ring structure. Right. Now let's talk about the second component beside base. The second one is sugar. So sugar that we present in nucleic acid is a pentose sugar, means it has 5 carbon ring, right, or a 5 atom ring. So this is a pentose sugar over here. So if you see carefully, this is a pentose sugar that we only learn in carbohydrate chapter, right? In RNA, ribose is present as a pentose sugar, but in DNA we have deoxyribose. Why is deoxyribose in DNA? Why the name is deoxy, whereas RNA is just ribose? So if you see the difference on the second carbon over here, in ribose you have OH, hydroxyl group, but in DNA on the same second carbon you don't have any oxygen. Oxygen is missing on the second carbon of this pentose sugar. So that's why if you are missing, D means removing, oxy means oxygen, that's why it's called deoxyribose again. So in DNA, pentose sugar doesn't have an oxygen on the second carbon, right? So in DNA with no oxygen atom on the second carbon, and then you will see the numbers are already given 1, 2, 3, 4 and 5 prime. So if you have anything 5 with a something this kind of line we call as prime. So that we call as 5 prime. This is 3 prime. Right. So if I say from now on this is a 5 prime and 3 prime, and you should be able to know that what I'm talking about. That means I'm talking about the 5th carbon or the 3rd carbon. If I say 2 prime, then second carbon, right, or second atom. So we already talk about nucleotide. Now we're going to talk about nucleoside. Right, so there's a difference between nucleotide and nucleoside. So what's the difference? In nucleotide you have three components. You have base, you have sugar, you have phosphate, right? But in nucleoside you just have base and sugar. You don't have any phosphate in nucleoside. So nucleoside is a combination of only sugar and base, and it's normally produced when the nitrogen atom in the pyrimidine form a glycosidic bond to the first prime of sugar, right? Whether it's ribose or deoxyribose. So let's see the next. So this is your ribose in RNA, because ribose present in RNA. This is the first prime carbon, and this is your base, right? One of the base, which is purine, right? One of the purine, which is adenine right now, A. Right? So in adenine, the hydrogen from the adenine from the nitrogen combined with an oxygen from the carbon number 1 of the sugar molecule and remove as water, if you can see over here. Now if you remove H and OH, what's gonna left? The nitrogen is going to attach directly with this carbon number 1, and if you can see this bond over here. And then we know that if any carbohydrate make a compound, we call as, or a bond, we call as glycosidic bond. We talk about this bond in carbohydrate chapter. And this is beta in glycosidic bond. Why? Because this OH is on the upside, so this is a beta isomer. This sugar is a beta isomer of ribose, right? And this hydrogen comes from the nitrogen, so the nitrogen is attached with first carbon, so that's why it's beta, and N for nitrogen, so that's why it's beta in glycosidic bond, right? So this is the reaction when sugar and base combine, you're gonna make nucleoside. So this is your nucleoside, sugar and base together, sugar plus base joined with the help of glycosidic bond, right? And we already talk about the nucleotide. Let's talk about one more time, as we already know about it. Nucleotide is made up of a base, sugar and phosphate. So on the previous slide we already talked about how base and sugar normally combine together, but in this we're gonna see how phosphate is gonna combine. So these two get together is nucleoside, SIDE. So if I join phosphate group with nucleoside, we're gonna make nucleotide, right? So this is your phosphate group, right? And present in ionic form at a cellular pH. Whatever the pH of the solution that we have within the cell in living organism, phosphate is present in ionic form. If you can see, this is negative negative, and you have a phosphate, PO4 2 negative, right? So that means phosphate has a -2 charge, right? So now let's see, we already talk about, this is your sugar, right? This is sugar, this is your base, and this is your phosphate. So the hydrogen from the nitrogen react with the first carbon of the base of the sugar and remove as water. So this is a nucleoside, but the same sugar molecule, fifth 5' carbon, this carbon also has OH, it bind to the phosphate OH and remove this as water. Now all the three is now connected. So this is one bond, that new bond formed, and this is the second new bond that's formed, right? So this is a nucleoside. This entire thing is nucleotide. right? And I already told you, because of the phosphate, this 2 negative charge, this nucleotide has a negative 2 charge. Right? Okay. So how the nucleotide form, base is always attached to the 1' position of the sugar, that I already told you. This is 1'. Phosphate is located at the 5' position, which is this is 5' carbon of the sugar. So phosphate is going to attach to 5' always. This is how you make nucleotide. Okay, so this is the general structure of nucleotide. You have sugar, you have base, you have phosphate. All three are present, right? And over here, if you see, this is the second carbon, this is first carbon. So this sugar is deoxyribose. If you remember, because on the second carbon you don't have any oxygen, you have just hydrogen, right? But if it was only ribose, then the OH should be present in there, but OH is not present, only hydrogen is present. That means this is deoxyribose. And the time we know this is deoxyribose sugar, we know this nucleotide belongs to DNA, because we use deoxyribose. So this nucleotide present in DNA, not in RNA. Right? And you can also draw the nucleotide like this: base, sugar, base attached to the sugar, and phosphate also attached to the sugar. So this is also nucleotide. Okay. So now let's talk about the component of DNA and RNA. Bases. We have 4 bases in DNA: A, G, C, T. Whereas in RNA, T is get replaced by uracil. Every other thing except same. Sugar, we have deoxyribose in DNA, in ribose in RNA. Nucleoside, base plus deoxyribose. This is the sugar, right? If it's DNA, then it should be deoxyribose, the deoxy sugar. In RNA, we have ribose sugar, base plus nucleotide. We have phosphate one attached to the base and deoxyribose. In RNA, we have base, ribose, and phosphate. And then nucleic acid, DNA, like I already told you, DNA, nucleic acid is a polynucleotide, where lot of nucleotide molecules combine together to form a polymer, right? So in DNA, this is a linear chain of deoxyribose nucleotide, because in this nucleotide we have a deoxyribose, this pentose sugar. Whereas in RNA we have ribose nucleotide, where sugar is a ribose molecule, right? So that's the only difference. Right. So identify the following base as your purine or pyrimidines. So if you have adenine, it belongs to purine or pyrimidines. So you have to see. And then for cytosine, whether it's present in purine and pyrimidines. So if you can see, adenine belongs to the purine. Why? Because it has 2 rings, and we know purines is adenine and guanine, Ag. And cytosine belongs to pyrimidines because it has 1 ring structure, and pyrimidines has cytosine and thymine, right, in DNA. Okay, so now let's talk one more problem over here. Identify each of the following bases as your component of DNA, RNA, or both. So guanine present in both DNA and RNA. Adenine also present in DNA and RNA. Thymine is present in DNA only, not in RNA. Uracil present in RNA. Right. So this is how you normally identify which group or which bases are present in which nucleic acid. right? So remember this thing, uracil always present in only in RNA, whereas thymine is present in DNA, right? And thymine and uracil get interchanged. In DNA you have thymine, in RNA you will have uracil, and all other three bases are same: cytosine, guanine and adenine. These three bases are present in both, but only thymine and uracil get replaced between the two different nucleic acids. Right, so this is the answer that we already talked about. Now let's talk about the primary structure of the nucleic acid. Like we have primary structure of proteins where amino acids are joined together through peptide bonds. Similarly, in nucleic acid, in the primary structure of the nucleic acid, we have a polymer of nucleotide. Polymer of nucleotide, where many nucleotide comes together and bind through a bond to make polynucleotide. Polynucleotide, right? So let's take an example over here. You have base, you have sugar, you have phosphate group. So base, sugar, phosphate, this is one nucleotide, right? Then again you have second base, sugar, then again phosphate. This is your again nucleotide, right? And then you have one more nucleotide over here, and then you have one more nucleotide. So this means this is made up of 4 nucleotide, and then these nucleotide is gonna bind with one special bond called phosphodiester bond, right? So we're gonna see in the other slide what are these phosphodiester bond is and how this nucleotide bind together and form a polynucleotide, right? So nucleic acid, like I will tell, as a polymer of many nucleotide, have a unique sequence of bases which is called their primary structure. In proteins we have a unique structure of amino acid. What are the sequence of amino acid? If the sequence of amino acid change, the primary structure of the protein change. Similarly, in DNA we use a sequence of bases. What are the bases? Cytosine, guanine, thymine, adenine, uracil. So we're gonna see what are the sequence of these bases in any primary structure of nucleic acid. If this sequence is changed, that means you have a different structure, primary structure of the DNA, right? So we're gonna see how we use this unique base sequence. So let's talk about how this nucleotide bind together, because nucleic acid is a polymer of nucleotide, right, which is polynucleotide. So let's talk about this is one nucleotide. Why? Because you have base, you have sugar, you have phosphate, right? This is your second nucleotide. You have a base, sugar and phosphate. So when these two nucleotide comes close together, we're gonna have some special reaction going on. So what's the reaction? The 3' end of the sugar, the third carbon of the sugar and the OH bond attached to this sugar is going to react with the phosphate from the second nucleotide, right, in the presence of acid or H+ ion. So what's gonna happen? Then this hydrogen and this hydrogen combine together and react with, so this H+ ion and this OH comes out as water, and then this O is gonna attach with this carbon, right? Because this bond is going to break, which is bound to the OH, because this OH is going to bind with H+ ion and then remove as water. So let me do one more time. So this bond is going to break and this OH and H is going to make up combine together and make water as a product, which is this, right? And then now this carbon need one more bond, because carbon need 4 bond, then oxygen is gonna come and bind with this carbon, and now you will see this oxygen from the phosphorus is bind to this third carbon, right? This is how you normally join two nucleotides together. The OH group from the third carbon of the sugar molecule is going to attach with the oxygen, one of the oxygen of the phosphate group, right? And now this type of linkage we call as phosphodiester linkage. Means you have phosphate group and you have two ester linkage. So this is one ester linkage, right? And then this is second ester linkage. So you have two ester bond between two nucleotide molecule. That's why it's called phosphodiester linkage, right? So make sure you remember what kind of bond normally being used to join two nucleotide molecule together is a phosphodiester bond. And one more thing, important thing that you need to know from this structure, if you see the top nucleotide over here, you will see the 5' end of the carbon, as we know, attached with the phosphate group, right? So we already learned about this thing. So this phosphate group is free. This phosphate is not attaching with any other nucleotide, with the any other nucleotide, right? Because we don't have any nucleotide on the top. So this phosphate is free. So that means we call this phosphate as free 5' phosphate. So that's why for this nucleotide this is a 5' end, right? And if you go to the second nucleotide, the bottom nucleotide, right? So over here this 3' carbon of the sugar still has OH group, because we don't have any more nucleotide underneath it so that it can bind to this oxygen, right? We don't have any more, so this is free. If it was one more nucleotide, it's gonna bind, this carbon is gonna bind with that nucleotide after removing this OH, but right now that we don't have any nucleotide, so that means this OH is free. So this is we call as 3' end. So 3' end is that end where you have the free OH group attached to the third 3' end, 3' carbon of the sugar, whereas 5' end is where you have the free phosphate group, right? 3' and free 3' hydroxyl group. So from now on, if I say 5' end of that nucleotide, we are talking about the phosphate group. If we're talking about the 3' end of the nucleotide, we are talking about the free 3' hydroxyl group, right? So now let's talk about the nucleic acid sequence, the primary structure of the nucleic acid, right? So in this particular structure we have 4 nucleotides. 1, then we have 2, then we have 3, then we have 4. We have 4 nucleotides together because each of them has base, sugar, phosphate, base, sugar, phosphate, base, sugar, phosphate, and base, sugar, phosphate. So each of them belongs to one nucleotide. And how they are attached to another nucleotide? If you can see, all the 4 nucleotides, 1, 2, 3, 4, all the 4 nucleotides are attached through phosphodiester bond. Second phosphodiester bond, third phosphodiester bond. So to attach 4 nucleotides you need 3 phosphodiester bond, right? And then you have the top nucleotide that has a free 5' end, and then the fourth nucleotide has a free 3' end hydroxyl group, because we don't have any nucleotide below than this and we don't have any nucleotide above than this. That's why they are free. They cannot make any more phosphodiester bond, right? Okay, so this is the structure of nucleic acid, right? So now talk about what are the bases we have. We have adenine in this particular thing, we have cytosine, we have guanine, and we have uracil. The time I say uracil, I know that we are talking about RNA because DNA doesn't have uracil in this primary structure, right? It should have thymine if it was a DNA, but this is uracil, so that means this is RNA molecule that we are talking about right now. So so like I already told you, the sequence of the base is very important, right? Was the first base, second, third, fourth, and we always read DNA from 5' end to 3' end. We always read from DNA or any nucleic acid. Like protein, we always go from N terminal to C terminal. Similarly, in nucleic acid, we always read DNA molecule or nucleotide from 5' end to 3' end. So let's talk about on the 5' end which base is present. Adenine. So we write adenine over here. This is the sequence. Then we have second cytosine. Then we have C cytosine here. Then we have guanine, G, then we have uracil. So if nothing is written, that means this is 5' end, this is 3' end, right? And we always read from left to right, 5 to 3, A, C, G, U, right? So make sure you know this concept, right? Okay, so now let's try to do one problem. In a nucleic acid, the nucleotide are joined by which kind of bond? Phosphodiester bond. The answer is phosphodiester bond we have, where two nucleotides normally get joined with the help of this bond. So now let's take one more example. In the nucleic acid CGAU, which base is at the free 5' phosphate end? So we know whenever we read any base sequence in a nucleic acid, we always read from 5' to 3' always. So it's asking which base is present at the 5' end. So this is the base that is present at the 5' end, which is cytosine, right? So the answer is A, cytosine, right? Even though nothing is written, you always assume this is 5', this is 3', right? So now we're going to talk about what is DNA double helix, what's the structure of the DNA, and what is the process called replication is, right? So before we go to the DNA replication and what's the structure of the DNA, we talk about how the structure looks like. We're going to talk about first what are the DNA base pairs is, right? So previously during the 1940s, some of the biologists determined that the bases in DNA had a specific relationship. Means they always bind to a spatial basis. So one base always present in equal amount to another base. What does it mean? They found out that the amount of adenine was equal to the amount of thymine in the DNA. If you have a DNA molecule, then if you have two adenine molecules present, that means you should have two thymine molecule also. They have to be equal. And the amount of guanine is always equal to the amount of cytosine. So they are equal in number, right? And they also figured out or found out that adenine is always get paired with thymine. That means A and guanine is always paired with cytosine. Means whenever you have an adenine, it always get paired with thymine. Whenever you have cytosine, it always get paired with guanine. But they cannot get paired with different bases. A cannot bind with G, C cannot bind with T. So they always be A and T together. A always bind with T, C always bind with G, right? So that means the number of purine bases is always equal to the number of pyrimidine bases. Why? Because what is purine? A and G. If A is always equal to the thymine and C is also always equal to the number in cytosine, that means number of purine bases is always equal to the number of pyrimidine base, right? So because they are equal, right? So remember this thing. In any DNA molecule, you will always have this, or mostly have this type of information. Means A always has an equal amount T and G always has an equal number to C. Now let's talk about the DNA structure. There was two scientists called James Watson and Francis Crick. They proposed in 1953, they proposed that DNA was a double helix structure. Means they are double helix, something like this. What is double helix? Consisted of two polynucleotide strands winding about each other like a spiral staircase. What does it mean? So this is your one polynucleotide, right? This is 5 prime end, this is 3 prime end, because this is a polynucleotide, and this is a second polynucleotide, right? So this is 3 prime end, this is 5 prime end. So DNA is a double helical structure. Double helical. If you can see, you have two helical structure, one from each polypeptide, and they wind together, right, within itself. And it contains sugar phosphate backbones along to the outside steering rail with the base arranged like a step inside. So what does it mean? So let me show you over here. So this is one polynucleotide, and from this is 5 prime end, this is 3 prime end, and this polynucleotide is made up of what? Base, sugar and phosphate, right? So this sugar and phosphate is like a backbone. So this backbone is made up of sugar and phosphate, and the bases are coming outside from this polypeptide. This is the base. So it's look like that look like a stair railing. This is the outside of the stair railing, right? And this base is looks like a step along the railing around the stair, right? This is look like a step. So always remember this: in DNA double helical structure, the backbone is formed by sugar and phosphate. Backbone, right? And the bases are present or coming out from the polynucleotide chain. So what does mean? It looks like something like this. This is base coming from the green polynucleotide strand. This is base, and similarly red also have bases coming out, right? So the bases are present within the polynucleotide chains, right? Like a stairs. And one strand has normally goes from 5 prime to 3 prime. Like this is first strand, it goes from 5 prime to 3 prime, whereas the second strand goes from 3 prime to 5 prime. This is second strand, 3 prime to 5 prime. If you can see the top side, that both polynucleotides are anti-parallel. What is meant by anti-parallel? Anti-parallel means both polynucleotides are parallel but they are in opposite direction. So one strand start with the 5 prime end, but the second strand that is just next to the top one is the 3 prime end. So you won't see the two polynucleotide has the same prime end on the same side. They're always in opposite way. If you have 5 prime end on one polynucleotide, you will have the 3 prime end on the second polynucleotide in a double helical structure. Similarly, on the bottom you have 3 prime end and 5 prime end, right? One's coming from the red polynucleotide, another comes from the green polynucleotide, right? So you have to make sure you remember this. They are anti-parallel, right? Okay. So now let's talk about the complementary base pair. What is meant by complementary base pair? Like we already talked about that adenine is always equal to the thymine and guanine is always equal to the cytosine, right? We already talk about this thing. Number of adenine molecule will always be equal to number of thymine and guanine is always equal to the number of cytosine. So now we're gonna see why this is the case, right? DNA contain complementary base pair, equal amount of A and T and equal amount of G and C, in which adenine is always linked to thymine molecule. So that means adenine always bind to thymine, always binds to thymine, and guanine always bind to cytosine. You have to make sure you know this thing. And then how they normally bound? Adenine and thymine are bind through 2 hydrogen bond. So that means I can write something like adenine 2 bond thymine, and these are hydrogen bonds, right? Similarly, guanine always bind to cytosine through 3 hydrogen bonds. So that means G has 3 hydrogen bonds, right? And then you can also see the structure. Through structurally you can say this is one of the hydrogen bond, this is second hydrogen bond between A and T, right? Similarly between G and C you have 3 hydrogen bond, 1 2 3. So that means if your DNA molecule have high number of GC, or we can say if you have high number of guanine and cytosine in your DNA, that means your DNA is strong because you have 3 hydrogen bond compared to if you have high number of adenine and thymine, because adenine and thymines are bind through only 2 bonds, right? So if your DNA molecule, which is a double helical structure, if your DNA contain high number of GC content, that means you have more number of hydrogen bond because each GC has 3 hydrogen bond. If you have high number of GC in your DNA, that means the DNA is stronger. You need more energy to break 3 bond at a time compared to 2 bond between adenine and thymine, right? So this is how you normally determine the strength of the DNA molecule. So now let's talk about the double helix. Double helix, we already talked about, has 2 strands of nucleotide that binds together, and it normally stays like this because of the hydrogen bond. A and T has 2 hydrogen bond, G and C has 3 hydrogen bond. Because of this hydrogen bond, this double helical structure is possible. Or stable, right? If there was no hydrogen bond, this double helical structure will unwind into a separate nucleotide, right? But because of hydrogen bond, they form double helix structure, right? So this is the structure, right? You have one helical structure, you have second polynucleotide, and you can see this is 5 prime end, then this has to be 3 prime end, and you have the bases going inside from both the nucleotide. So A is always bind to T with 2 hydrogen bond, and G always bind to C with 3 hydrogen bond, right? And this backbone, this railing, looks like railing, is a sugar phosphate backbone, right? So now let's talk about the complementary base sequence of the matching strand in the following DNA sequence. So what does it mean? Let's assume we know DNA is a double helical structure, but right now we are not making a double helical structure. We are just asking that if one of the sequence from 5 prime to 3 prime has this sequence A, G, T, C, A, T, what should be the sequence on another strand? So another strand has to be 3 to 5 prime, because on the same side you cannot have the same prime, right, in a double helix structure. So even though they are double helical, but in this, for this particular problem or this type of problem, we are assuming you have this sequence. So you have to just fill out the sequence on the opposite strand, right? And we know the bases are complementary, because we know A always bind with T and G always bind with C, right? We know this thing. So let's try to make the base sequence on second strand. If the first strand has A, the second should have T if it is a DNA molecule. This is important. If it was RNA, then it's gonna be U, because in RNA you cannot have T, right? But this is DNA. T, G, C, T, A, G, C always bind with G, C always bind with G, then T, T, then A, then G. So this is a sequence on the other strand of the molecule, 3 prime to 5 prime, right? So that means if I know the sequence of one of the strand out of the 2 double helical structure, then we can easily figure it out what's the sequence on the second strand of the double helical structure, because they are complementary. And we know if A is present on one strand, then you should have T on the other strand, just opposite to it, right? So this is the answer that we already talk about. Talk about the DNA replication. What is DNA replication? Why cells need to replicate its DNA? So we know cells normally divide to replace the dead cells. So cell has to do cell division, divide into 2 to replace all the dead cells that we have in our body, right? But before the cells divide, they have to duplicate all their content, including the DNA, because once they divide, it has to distribute equally all the content within the 2 cells, right? So DNA replication purpose is to duplicate the DNA molecule that we have within the cell before the cell division, right? So that they can distribute equal DNA to both the new daughter cells that is produced by dividing the parent cell. Okay, so now let's talk about what is DNA replication. DNA replication is making a copy of the DNA, right? Where you're making exactly the same copy of the DNA. So to make a copy, let's say if this is your DNA, parent DNA, this thing is your parent DNA. If you want to copy this parent DNA, what you need to be done first, you have to open this double helix, right? Because if you don't open the double helix, then you cannot get the bases to read, because the information is present in the form of bases A, G, C, T. If you cannot open the double helix, how you going to get and copy all the information, right? So first step is to open up the double helical DNA. So this function is done by an enzyme called helicase. So this helicase catalyzes the unwinding of the double helix DNA by breaking the hydrogen bond. We know the bases are attached together through hydrogen bond, and because of this hydrogen bond, the double helix structure is possible for the DNA. So helicase normally open up by breaking all the hydrogen bond between the bases. If the hydrogen bond is broken, then the double helix is going to open up easily, right? And once it's open, so this is your parent DNA, right? This chain is come from parent DNA, and this chain of polynucleotide also comes from parent DNA, right? And once the DNA is open up, now you're gonna see a new chain is being formed by using all the information from the parent chain to daughter DNA. This is called daughter chain, or new DNA, you can say, new DNA. Similarly, from the other strand of the parent DNA, you will form one more DNA or nucleotide sequence, right? That is using all the information from the parent chain. So if this parent chain has adenine, then daughter or the new DNA should have thymine, right? If you have guanine in the parent chain, then your daughter should have an cytosine C, because DNAs are complementary, right? Similarly, over here, if this is thymine, then your new DNA should have adenine in there corresponding to thymine, right? This is how the information is being copied into a new DNA. And then this copying of information or making of this new DNA is done by one more enzyme called DNA polymerase. So if I ask you which enzyme normally has been copying the DNA, is DNA polymerase. Which enzyme is normally helping to open up the DNA is helicase, right? So once the DNA replication is complete, you will see 4 DNA, right? 4 DNA chain. 1, then you will have 2, then you will have 3, then you will have 4, right? And then you will see, you will get 2 copy of DNA. This is 1 copy, this is second copy, right? This is a double helical structure again. You have 2 nucleotide together, right? These 2. So that means this red one comes from parent. This new one is a new DNA. Similarly, the second pair of DNA contain 1 DNA from parent, from the original molecule, and the new one from the new DNA, right? If you can see over here, 1 from parent, 1 from new, 1 from parent, 1 from new, right? So that means DNA replication is semi-conservative. What is meant by semi-conservative? Means each daughter cell after division will get 1 part from the parent DNA and 1 part is a new synthesized DNA, right? Something like this. Right. So if you see, the daughter cells usually get 1 DNA from the parent chain and 1 from the new DNA that is formed. So that means DNA replication is semi-conservative, means each daughter cell will get at least 1 part of the DNA from parent chain, right? So that's why you always get a genetic information from parent whenever you form a new cell. Right, so that's why it calls semi-conservative. This is very important. Okay. And one more thing, that what is the function of DNA ligase? So let's assume this is your DNA from the parent chain, and this is the new DNA that you are forming, right? New DNA or new polynucleotide chain or new DNA, you can say, right? So once it get formed, this is not forming as a continuous change. They usually adding a polynucleotide. If you remember, DNA is what? Polynucleotide chain attached together. So how they are going to get attached? Because in the previous slide we just show you a single line, right? But the DNA polymerase is not adding all the bases or nucleotide together as a single line and join, right? So the joining has to be done by a special enzyme called DNA ligase. So DNA ligase normally joins nucleotide together. So that means once the first nucleotide get added onto the new DNA, and then DNA polymerase add the second nucleotide, then the 2 nucleotide is going to add by DNA ligase, right? So DNA polymerase and DNA ligase is getting work together, right? So this is the function of polymerase and DNA ligase. It's like a glue that add up all the nucleotide together, right? Okay, so now let's talk about One of the question, how many hydrogen bond link in each of the following base pair? If you have adenine and thymine, how many hydrogen bond? 2 hydrogen bond. If you have guanine and cytosine, how many? 3 hydrogen bond. Right, so you have to make sure you remember this. Right, 3 and 2, we already talk about this thing. Now let's talk about RNA and transcription. So till now we talk about DNA. What is DNA? What is the structure of DNA? What is DNA replication? Now we're gonna focusing on our talk on RNA and what is transcription. So we already talk about RNA is ribonucleic acid and it make up most of the nucleic acid found in the cell, right? And it transmits the genetic information needed for cell operation. We're gonna see later in the chapter how this RNA is normally helpful to transmit the genetic information from the nucleus to the outside cell of outside nucleus, right? To do a cell operation. Okay, and then we already talk about RNA is a molecule of polymer of nucleotide and it differs from DNA in 4 ways. The sugar in RNA is ribose. We already talk about in DNA is deoxyribose. In RNA we use uracil. In DNA we use thymine. And the most important thing is mostly RNA molecules are single stranded, not double stranded. Whereas DNA is a double stranded strand most of the time. RNA molecules are much smaller than DNA molecule. So there are these four differences between DNA and RNA. So now let's talk about what are the different types of DNA that we can find in the cells. So there are 3 different types of DNA you can find in the cell. Like I already told you, RNA transfer information from DNA and to make proteins, right? So we'll see about later on the slide how it's possible. But let's talk about what are the 3 different types. The first type is messenger RNA, which is also called mRNA. Second one is transfer RNA called small t RNA. T is for transfer. The third one is ribosomal RNA, small r for ribosome and RNA. And let's talk about what are their function is. Messenger RNA carries genetic information from DNA in the nucleus to the ribosome in the cytoplasm. Transfer RNA interprets the genetic information in the mRNA and brings specific amino acid to the ribosome for protein synthesis. Right. And ribosomal RNA is the most abundant type of RNA and is combined with protein to form ribosome. So let's talk about these things individually so that you can better understand what's the function of each of them separately. So let's take one example. This is your cell. This is your nucleus, and within the nucleus what you have? DNA. Right. So what is mRNA? And this is cytoplasm. Right. And this is nucleus. Right. And then in nucleus what you have? DNA, right? So as we know, all the information that cells need to do its function is present in the DNA molecule in the form of base sequence. So all the information is present which enzyme need to activate, whether cells need some oxygen or carry out some reaction. Every information is contained within this DNA molecule. But let's say if the cells need to produce an enzyme, this DNA molecule has to release information outside the nucleus, right? To tell that part of the cell that produce protein that we need this protein and you need to make a protein. But the problem is DNA cannot cross nucleus. DNA cannot cross and come to the cytoplasm where mostly the protein synthesis occur. So how does the information is going to transmit from DNA to cytoplasm? Because DNA cannot cross this nucleus membrane and cannot go into like by itself outside the nucleus, right? So this information is being transferred with the help of messenger RNA, as the name is saying, messenger. So the DNA tells messenger RNA that you need to go outside the nucleus and then tells the protein machinery to make protein, right, or any other information. So messenger RNA carries genetic information, right? It takes the order from the DNA and then come outside from the nucleus. So DNA form mRNA, and this mRNA can cross the boundary of nucleus membrane and go inside the cytoplasm, right, with all the information that DNA wants to deliver, right? So this is also carries genetic information. Transfer RNA interpret the genetic information. Now the mRNA that has the information needs to be interpreted. Somebody needs to read this information, that what's written in the information, so that they can start working. So this interpretation is done by transfer RNA, right? And then ribosomal RNA is a place or is an area where protein synthesis occur, right, as an area or place, you can say, right? So ribosomal is the most abundant type and combined with proteins to form ribosomes. So ribosomes we also call as a factory. It acts as a factory where all the information goes inside and the protein is normally formed, right? It's like a factory where everything is normally made once you have every information that you need to make something, in case proteins, right? Okay, so this is the same information that we talk about RNA, messenger RNA, right? So RNA is has the highest percentage of the total RNA, right? Is majorly present RNA in the cell, RNA, right? So tRNA, let's talk about a little bit about the tRNA. This is a transfer RNA and it's read the genetic information, right? Right, so mRNA has all the genetic information, and then this tRNA goes to the mRNA and read all this information, right? We're gonna see in protein synthesis how this tRNA works and read the information, but right now just remember this thing. This tRNA has a special area called anticodon over here, which is a series of 3 bases that complement 3 bases on mRNA. So this is the area that normally helps to bind with mRNA, that complement with 3 bases of RNA, means they are complementary to the mRNA, so they bind to the mRNA molecule, this tRNA molecule, right, because of presence of anticodon, right? So make sure you know, if I say anticodon, you make sure you know it's present on the tRNA. So now let's talk about the flow of genetic information, how the genetic information normally flows within the cell. And this is also called central dogma of molecular biology, right? So what is central dogma? Is a process by which genetic information stored in the DNA molecule is expressed in the structure of synthesized protein. Means whatever the information DNA has, this is DNA, it converting into proteins, and we know proteins are enzyme and they do many function within the cells, right? So DNA direct the synthesis of certain protein, and each protein in the body correspond to DNA. So there are 2 steps how DNA information converting into a protein, there are two steps. One is called transcription. Transcription is a process by which the genetic information is being transferred from DNA to mRNA. Right? So transfer of genetic material from DNA molecule to a molecule of mRNA. So the formation of mRNA from DNA we call as transcription. And now this mRNA can cross nucleus and go to the cytoplasm. We already talked about this thing. Right? Once the mRNA is in place in the cytoplasm now outside the nucleus, then we begins protein synthesis. Then the second process comes into play, which is translation. Translation is a conversion of the code carried by messenger RNA into an amino acid sequence. So what does it mean? This is the mRNA that has all the sequence or all the information from the DNA, and this information is converting into amino acid sequence, which is we known as protein. Proteins are amino acid polymer, right? And the mRNA has information in the form of bases A, G, C, U, right? G, C, something like this, right? And this information is normally decoded in the form of amino acid. We're gonna see how, but this process we call as translation. Right? So make sure you know the difference. Transcription is formation of mRNA from DNA. Translation is formation of protein from mRNA. So central dogma means this is DNA. If you do a replication, if you make many copy of the same DNA, we call as DNA replication. But if DNA goes transcription, that means they are making mRNA. And once you make mRNA, then the second process called as translation, that convert mRNA into a protein. Right? So this is a flow of genetic information. How does DNA normally send the genetic information, right, to the cell? So this is an overview of the protein synthesis. Right, the genetic information DNA is replicated in cell and used to produce mRNA code for amino acid needed for protein synthesis. So this is just an overview. Like I told, this is nucleus, and within nucleus what you have DNA. Once you do transcription, then the DNA information is converting into a mRNA sequence. So it has all the information that DNA wants to deliver outside the nucleus. So the cells can use this information to carry out the function. So the mRNA goes outside the nucleus and present in cytosol, which is also called cytoplasm. Right? And this mRNA, once present in the cytoplasm, goes to a machinery or factory called ribosome. Right? It goes to the ribosome, which is called factory, protein factory. And within the ribosome, you make protein through the process of translation. Right? So you will see amino acid sequence. This is right, polymers. Or you can say polypeptide or proteins. Right. So let's start with protein synthesis first. Right? The first step in protein synthesis is transcription. And we know transcription is from DNA to mRNA. Right? Transcription begins when the section of DNA containing the gene unwind and RNA polymerase enzyme use one of the strands. So what does it mean? Let's say this is your DNA, double helical DNA, right? And this DNA wants just this part of the DNA to get copied and or send this information. So whatever the information DNA wants to send, it present in this small part. Let's take an example. So the region, this double helical open up from this end, this area, and then this DNA that has all the information is open up. Once the DNA information is open up, as we know from DNA replication, the bases has all the information, and to copy the information you have to open the DNA so that the enzyme can copy the information, right? So in this case we are making mRNA, so that's why we are going to use RNA polymerase. So to make an mRNA molecule from DNA, we use RNA polymerase. So mRNA is synthesized using complementary base pairing with uracil pairing with adenine in DNA. So let's assume this is the required or the region of the DNA that has all the information that the cells need. So the double helical open up, expose all the bases over here, and then this RNA polymerase start copying this information. So it start from here. So if DNA is A, then mRNA will have U. If you remember, RNA cannot have thymine, so A always bind with U in RNA. So make sure you know this difference. If it was a DNA, then it should be T, but we are talking about mRNA, then it should be U. Thymine with adenine, guanine with cytosine, so you will see a complementary base pairing in mRNA, right? So once the mRNA is formed, what's going to happen? This information is going to go outside the nucleus, right? So what is RNA polymerase? RNA polymerase, we already talked about, is the enzyme that helps to copy the DNA into the mRNA molecule, right? And the region that has the information is usually open up, right, so that the RNA polymerase can copy it, right? So this is a process of transcription, DNA to mRNA, with the help of RNA polymerase. This is the enzyme, right? So there are two types of cells called prokaryotic cells and the eukaryotic cells. So we're going to talk about only eukaryotic cells in this chapter, and eukaryotic cells belongs to humans, right, or living organism mostly that we have, basically humans. So in humans, the segment of the DNA are interrupted by introns. So let's say this is DNA segment, right, and it has the information that the cells need, right? But the problem is over here in this DNA region that need to be copied in the form of mRNA contains some spatial region called introns, right? And you have some other region that is useful. This green one is a useful region which is called exons, that has the information actually that is present, that gives the actual information. Introns doesn't carry any information. It's like a waste material that is present in the DNA. When you copy DNA information in the form of mRNA, we don't want any intron, right? But DNA carry intron and exon that carries information. So when RNA polymerase copy this information from DNA to mRNA, they also copy the intron thing, which is not useful. We don't want intron thing to come into the mRNA, right? So transcription occur in both intron as well as exon, because RNA polymerase doesn't know which area of the DNA, whether it's exon or intron, which area is useful and or which area has the information, right? So they just do the transcription for entire DNA thing, right? Which leads to the production of heteronuclear DNA, RNA. What is heteronuclear RNA? Is that RNA that is made from the DNA molecule directly, that is made up of intron and exon. So let's take this example over here. This is your DNA. It has an exon which has the information and introns that doesn't contain any information. But during transcription, RNA polymerase copy everything, and in mRNA molecule called heteronuclear RNA contain information from exon, intron, right? But intron doesn't contain any information, so we don't want in RNA molecule any of the information from intron. So after you made the heteronuclear RNA, some other enzyme comes and then just clean up this intron and take out this intron molecule from the heteronuclear RNA and then join the green part together, which is useful, which is exon, right? So this useful part we call as actually mRNA, that has all the information without getting interrupted by introns, right? So make sure you understand this thing. DNA form heteronuclear RNA because DNA is an intron, and then during transcription this intron also get transcripted into a RNA molecule, right? But we don't want this intron. They just cleaved up after you make a heteronuclear RNA, and then all the green exon part get attached together through enzyme, and this spatial part we call as mRNA, this spatial molecule, right? So mRNA contain only the useful information. From DNA, right? Okay, so now let's talk about this particular problem. What is the sequence of the bases in mRNA produced from a section of the template of DNA? So if this is your DNA, this and this thing is your DNA, what should be the mRNA sequence? You have to figure it out, this thing. So we know if we talk about mRNA, adenine is bound to uracil, right? So now let's go. If you have C, then it's bind to G, T bind to A bound to U, because we don't have T, right, in the RNA, then C, then C. So let's compare which one is this, G, A, U, U, C, C. So option B is your correct answer, because this T cannot possible in RNA, right? So option B is your correct answer. The answer is already provided over here. Now we're going to talk about the genetic code, and we'll see what is genetic code and how the genetic code helps in the protein synthesis mechanism. What is genetic code? Genetic code consists of a series of 3 nucleotides or 3 bases, you can say. Right. In mRNA molecule called codons. So what is genetic code? Genetic code is the sequence or consists of series of 3 bases or 3 nucleotides in mRNA called codon that specify the amino acid and the sequence in the protein. Means if this is an mRNA molecule, let's say this is mRNA and this is the sequence that you have in mRNA. You have uracil, all these uracil. So these 3 bases together form one codon. This 3 again, one another codon. This 3 again, another codon. So codon is nothing, it's just the sequence of 3 nucleotides together, right? And we'll see how this codon is converting into amino acid. How UUU normally called for one amino acid, right? Phenylalanine, right? And this process we call as translation, right? The conversion of mRNA sequence or mRNA information into. So this conversion of mRNA into amino acid sequence we call as translation. Then now let's see how this codon information can be translated into an amino acid sequence. So codon have been determined for all 20 amino acid because we have 20 amino acids. So for each individual amino acid we have a special 3 set of bases, right? Base, 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 that normally code for one particular amino acid, right? So let's go over here. So scientists have made a sequence of 3 nucleotides that normally correspond to one specific amino acid. So if you see how many bases we have 4 bases in our DNA. RNA, we have uracil, cytosine, adenine, guanine, and we have uracil, cytosine, adenine, and guanine, right? So if I have UUU, 3 bases together that code for phenylalanine, one of the amino acid. If you have UUC, again it code for phenylalanine. If you have CAU in your mRNA, cytosine, adenine, uracil, then it goes for histidine. So you can just check for each, you will see for each 20 amino acid you will have a specific codon assigned to it, means a sequence of 3 bases together, right? If you have GAU, then you have aspartate, right? If you have AAA, you have lysine. So depending on your mRNA, this is your mRNA, what's the sequence of the bases A, G, C, U, A. So once you see the triplet of the bases, if you have A, G, C, then you will have one amino acid assigned to it, right? And similarly you will see a triplet of bases, and then corresponding to that triplet bases you will have amino acid that code for that basis. Let's take a couple of examples. Determine the amino acid sequence from the following codon in the section of mRNA. So this is your mRNA, and you have the sequence CCU, AGC, CGA, CUU. So this is a triplicate. And now you have to tell me, if this is an mRNA sequence, if you do a translation process, what is the sequence of amino acid sequence, right? And then the codon information is already given. If you have CCU, that means that particular sequence or basis correspond or code for proline, right? And similarly you have all other different basis sequence or triplet. So let's use this information with your mRNA information. So first you have CCU. So if you have CCU with CCU, so this is your CCU, that means the first amino acid is gonna be proline, correspond to CCU. Then you have AGC, then serine, GGA is glycine, and CUU is leucine. So by looking at the base sequence, the triplet of the base sequence, we can easily see what's the sequence of the amino acid. Right? So if you have this mRNA sequence or information that comes from the DNA, then we can easily see what's the sequence of the amino acid. And from the amino acid and protein chapter, we know for protein to play a specific function, their amino acid sequence play a very important role. If you change the sequence, that means you are changing the function of the protein sometimes. So the sequence of amino acid is very important, right? Okay, so let's take one more example. So this is your mRNA, and you need to tell me what is the sequence of the amino acid. Once you do translation, this is the process, translation, right? So you have GCC, you have to see where GCC, this is GCC, that means the first one is aniline, then you have GUA, then you have valine, then you have GAC, then you have aspartic acid. So this mRNA sequence code for a special protein that has the amino acid sequence of aniline, valine and aspartic acid. That's from N terminal to C terminal, right? So this is the sequence that we already discussed about this thing, right? But let's assume one more example. Instead of this sequence, if we would have GCC, GAC and GUA, I just switch these two sequence, then the amino acid sequence will be alanine, aspartate and valine. Now these two proteins may be different because the sequence of the amino acid is changed, because the mRNA sequence is different. So everything is connected. mRNA sequence is different, that means new amino acid sequence. If you have new amino acid sequence, that means you might be producing a new protein that have different function than this protein, right? Because amino acid sequence are important. Now we're going to talk about the genetic mutation. What is genetic mutation is? So genetic mutation is a change in the nucleotide sequence of the DNA that may enter the sequence of the amino acid and affect the structure and function of the protein of the cell. So what does it mean? Let's say if this is your DNA, it has some sequence, right, in the form of bases, then this DNA is converting into, this information is going to convert into mRNA with the process of transcription, right? This is your mRNA now that has this information from the DNA, and now this information from the mRNA is used to form proteins or amino acids, which the process called as translation, right? So now you have amino acid 1, 2, 3, 4 and keep going depending on the sequence, right? So if we change any bases in DNA, let's say if I remove these two bases from the DNA, what's gonna happen? Now these two bases, if these bases are gone, then couple of bases from the mRNA will also be removed, because the transcription won't transcribe this empty sequence, right? And then if this information is removed from mRNA, then you might be leaving some of the amino acid from the proteins, right? And if you change the amino acid sequence, then you are changing the protein. That means the protein that you needed or required by the cell is not the same protein that the cell is producing, and this caused by mutation. You are making a non-functional protein, right? So that means if you change anything in the DNA sequence, you will change, the change will happen in the mRNA, and that change is going to convert into a protein that might be a non-functional protein, or that protein might not act as an enzyme that body required, right? And this is all because of the change in the nucleotide sequence of the DNA, and we call this as mutation, right? And this changing in the nucleotide sequence of the DNA may result from X-rays, overexposure to the sun like UV light, some chemicals called mutagens. Mutagens are those chemicals that can cause mutation, and some viruses are also responsible for this kind of mutation. So let's take an effect of mutation. Let's you start with DNA, and let's say you expose DNA with X-ray, UV light or chemicals or viruses that alter the DNA sequence. If you alter the DNA sequence, means you are altering the mRNA sequence. If you are altering the mRNA sequence, that means you are altering the protein sequence, or you are producing a defective protein now, because you might not have the right amino acid sequence. If you are producing a defective protein because of the mutation, that means that protein is not able to carry out the function of the required protein, and because of this you might have some genetic disease or cancer, because you don't have the right protein or right enzymes, because proteins are enzyme to carry out one important reaction that is helpful to remove any disease. Right. So alternately, if you have mutation, you can have genetic disease or cancer. Right. So it also says the protein that you may be forming through a mutated DNA may lose its biological activity. If the protein enzymes are not active, means you're not carrying that reaction that is important for body, and because of this you might be having some disease. Now we're gonna talk about the recombinant DNA. So recombinant DNA is the DNA of an organism that contains genetic material from another organism. What does it mean? Let's say this is your DNA. This is DNA, right? And then DNA from one organism. If this DNA is combined with another DNA molecule that comes from second organism, right? So this is we call as recombinant DNA. Right? And we need to use some of the enzyme to ligate or to add this second DNA to the original DNA. So what's the process? The process is, let's say this is your DNA, right? Circular DNA first, and then this is your required DNA, DNA2. So how you going to incorporate DNA2 in DNA1? This is DNA1 to make that recombinant DNA. So the first you need to an enzyme, you have to open it, you have to cut and open this DNA1, right? So what you need to do, you have to use a special enzyme called restriction enzyme. So restriction enzyme is like a molecular scissors. They cut the DNA. So they cut DNA. So once they cut, using a restriction enzyme, you will get this kind of DNA with a space provided, because this region of DNA that is been cut by the restriction enzyme is removed. And now what you're gonna get, you're gonna add this second DNA from another organism, and you're going to make a recombinant DNA, because this DNA has two genetic material, one from the first organism, second from second organism. So this is recombinant DNA. But once you put the second DNA into a DNA1, you need to join these two DNA molecule DNA, right? You have to glue it together. So the glue of a cell is DNA ligase. So DNA ligase is an enzyme that acts as a glue that joins two DNA molecules together, right? So DNA ligase used to connect DNA fragment during replication, or to put together pieces of DNA produced by restriction enzyme. So so recombinant DNA is normally formed by combining two genetic material together. With the help of, first you have to cut the original DNA to make a place for the incoming DNA, and then once the incoming DNA comes and join with the first DNA, then DNA ligase is going to join together both the DNA, right, and then form a recombinant DNA, right? So this recombinant DNA technology normally used to produce important medicines like human insulin, human growth hormones, malaria vaccine, right? That has different uses: protection against malaria, human growth hormone, treatment of dwarfism, human insulin, treatment with diabetes, right? So all these substances are made possible through the genetic engineering or through recombinant DNA technology.